Hi everyone, thanks for checking out this video. So this is going to be the next lesson in this series I'm doing on how to do basic transcriptomics in R. And so if you guys remember in the last video, we actually did like the basic differential expression analysis. And we came up with some lists of genes that were upregulated and downregulated in our IPF samples compared to the healthy control samples. So now in this video, I'm going to be showing you guys one way to visualize these results. So I'm going to be showing you guys how to make this type of plot here called a heat map plot. So basically this plot shows a selection of genes. So basically I've selected the top 100 most significantly differentially expressed genes between the IPF samples and the control samples. And basically when you make this kind of a plot, it basically makes a grid like this where it tells you in each sample kind of like how upregulated or downregulated the gene expression was. So if we take a look here, we have each row corresponding to a certain gene, and each column here corresponding to a certain sample. And we also have this bar up here color coding the class labels for each sample. So we can see the orange ones here are the IPF samples, and the yellow ones are the control samples. And then if we look on the grid and look for like a specific gene and a specific sample, we can tell how high the expression was of that gene in that sample based on how red or blue the color of this square in this grid is. So the more red it is, the more expression there was, and the more blue it is, the less expression there was. So for example, if we look at like this block of genes here, we can see that these were relatively lowly expressed in the IPF samples, because we see this block here, it's almost all IPF samples except for one. And we can see that for these genes, these samples had relatively low expression. But then we can also see the same block of genes had relatively high expression in these control samples over here, because we can see this block here is mostly red. And then for this kind of block in the middle, these are the samples where there was kind of a mix of high and low expression for these samples, and there's a mix of the sample classes too. And you can also note that these brackets here for both the rows and the columns are basically showing how the genes and how the samples cluster together in terms of how similar they are. So we can see with like this block of genes, since they're all kind of like clustered together in this bracket, that means that they're somewhat related to each other in terms of like having similar pattern of expression. And yeah, same with this block going down from here. We can see that since they cluster together in this bracket, they're kind of related to each other. We could say the same thing about the samples, depending on how they cluster together, but we don't want to draw too much of a conclusion from that, because remember, well, you guys are going to see, I haven't showed you this yet, but basically I picked these genes because these are the genes that have the most significant difference between control and IPF samples. So it's not surprising that the control and IPF samples mostly cluster together. That would be more of a notable result if you hadn't selected genes like specifically looking for the ones where that was going to be the case. Hopefully that makes sense. If that's confusing to any of you guys, just let me know in the comments and I'll try to explain further. But okay, so that's a little brief explanation. So now for the rest of this video, I'm going to be showing you guys how to make this plot. Okay, so let's get to it. So most of this is the previous code from the last lesson. If you guys have been following this series, the last lesson that I made was showing you guys how to do this differential expression analysis with this DESeq2 package in R. So if you guys haven't watched that video yet, I'm going to refer you guys back to that video, because this video is going to be building off of that already. So what I did just now before starting this video was I reran all of that code So now we have our R environment in the same state that it was in at the end of the last video. So if you guys aren't caught up with like the last video, I'm going to recommend that you guys go back and watch that, because you kind of need to have this code already run to do what I'm about to show you. So yeah, recommend you guys get caught up on that if you're not already. But okay, so now getting into the new stuff for this video. So the first thing is we need to load in this p heatmap package, and if you don't have it already installed, you're just going to run this command here and just run that in the command prompt down there to install p heatmap. That's going to be the package we're going to use to actually make the heatmap. Okay, and then the next thing is we need to decide how many genes we actually want to plot. Because if you guys remember, we got really a lot of significant results. There was a lot of differentially expressed genes between these two conditions. So if we just take a look at this, yeah, we got 7,822 differentially expressed genes. We can't really plot all of those because that'll just be too cramped of a plot, and we won't be able to see what's going on with them. Just be too many to try to plot. So we need to pick a number to focus on. So I'm going to pick 100. I'm going to take the 100 most significant differentially expressed genes to plot in my plot. But you guys can make this whatever you want. It could be like 200, could be 50, could be 20 even. But yeah, whatever you want to make it. Just I'm going to call this number n here, and then I'm going to say top genes arrow equals. And then I'm going to copy and paste in something, so just trust me on this for a second, but then I'm going to explain to you guys what each part is doing, basically. So you have top genes, and then copy and pasting in this. So yeah, let's start with the inside out, talk about what everything is doing here. So we're looking at the significant results table. So if you guys remember from the last video, this basically gives us this table with all the significant genes that came up in the gene expression, this like 7,822 genes. And then we have 6 columns telling us this information, including a couple different things. But what we're going to be interested in here is the adjusted p-value. So this is the p-value after doing this false discovery rate adjustment, basically. So yeah, what we want to do is we want to get a hold of this column here. So that's why we say $p adjusted. So yeah, that'll just give us that column. And then what we want to do is we want to get the order of these adjusted p-values from lowest to highest. So that way, our goal with this part here is to get this table ordered from most significant to least significant. In other words, in terms of lowest adjusted p-value to highest adjusted p-value. So yeah, we can do that like that. Now they're in order from most significant to least significant, and then basically what we're doing here is just getting the row names in this order, and then what we're doing with this final command head and then all of that comma n is we're getting the top 100 row names basically. This is just saying head like we want the beginning of this thing, and then n is how many we want. So this will give us the names of the top 100 most significantly differentially expressed genes here. So yeah, it gives us 100 here. And then yeah, then we're just storing those in this top genes character vector here. Okay, so those are the genes that are eventually going to be going into the plot. So now something else we need to do is we need to, before we make this heat map plot, we need to normalize the counts data. So I'm going to say norm counts arrow equals counts DDS, which remember is our DESeq data series, I think it stands for. Or no, sorry, DESeq data set object here. Remember, that's just our DESeq type object that stores all of our expression data, basically. So yeah, we can even just take a look at what we already have for this. So we can say like dim And yeah, just like our original data matrix, it has 18,838 genes, where every gene has its own row, and then 206 columns, where every column is a sample. Um, so what we need to do is we need to, because if you guys remember, we originally passed in unnormalized count data, but in order to make this plot, we actually need to normalize these counts to get it to look nice. Otherwise, if you don't normalize the count data, just the color scheme of the heat map won't really work out properly. Um, yeah. So we're just gonna say counts DDS and then normalize equals true. And then DESeq2 will kind of do its own normalization method, kind of internally here. So yeah, we can just run that. And then yeah, now it should look something like not integers anymore, but yeah. So now it looks like decimal numbers, basically floating point decimal numbers. So that's kind of a clue that it's normalized data rather than like integer count data. Okay, so what we want to do now is we want to select out the 100 genes that we're going to be plotting from this norm counts data matrix. So if you guys check the dimensions of this, it's still the same dimensions. This has the expression, the expression data, which is now normalized for all of the genes, not even just the differentially expressed ones, but all of them. So we need to get out the we need to get out the 100 genes that we're interested in plotting. So what we can do here is just say heat map data arrow equals norm counts, and then the row names of this are the gene names. So we can reference the genes we want just with their names, which is kind of nice. So we don't even need like the number indices here. We can just reference them just with the gene names because those are the row names we're looking for. And then we want all the columns, so we're just going to say just comma nothing here. Okay, so we can check the dimensions of our heat map data now. Should be 100 by 206. And yeah, we have 100 genes now. Every gene is a row, and 206 samples. Okay, looking good so far. And also, just to kind of check in on things, we can also just to check, print out the class of this heat map data, and yeah, basically just check that we're now dealing with a matrix here. This R data type. We're not dealing anymore with this like DESeq2 special object. Okay, so moving on, now we can begin actually setting up this plot. So now I'm going to begin actually like copy and pasting in some things. So the first thing to do for this heat map plot is just define the location we're going to be saving it, as well as like the shape of the image we want to output. And I made this kind of a large image just so nothing gets cut off or anything. But yeah, I'm going to save it in the plots folder, which reminds me we actually need to make that folder. So we're going to make in our workspace just some folder called plots. And now I'm just going to call it heatmapplot.pdf. And yeah, width 10, height 8. And wrap all that in PDF. Yeah. And then I'm gonna run all of this at the same time later, so I'm not gonna run that yet, although you could if you wanted to, but I'm not going to quite yet. The next thing to do, again, copy and pasting, being a little bit lazy here, so sorry about that, but yeah, I'm gonna copy and paste in. Yeah, so this part is basically defining the color scheme we want to use for the heat map. So I'm just setting up this color scheme because, like, at least my preference is that I want the heat map to be what it sounds like. Like, more expression should be red because it's hotter, and less expression should be blue because it's colder. So, I mean, you guys can make the expression colors wherever you want, but personally, that kind of makes sense intuitively to me because blue looks like a cold color, meaning there's like less expression, and R looks like a hot color. And it's a heat map, so we would think if there's like more expression, it would be hotter looking kind of, or at least that makes sense to me. Anyway, I'm also colorblind, by the way, just like fun fact or whatever. So I also just picked this because it looks like an easy color scheme that's not going to like confuse me too much. It was kind of funny. I saw one in like a paper a couple weeks ago where they made their color scheme like red and green, and I just wasn't able to tell the difference at all. So, so yeah, I had trouble with that. So I just make them blue and red because I can easily kind of tell the difference between them too. But yeah, this is just defining the color scheme like that, so that the lower expression ones are going to be blue and the higher expression ones will be red, and white is kind of like the intermediate. Okay, so next thing, again, going to copy and paste a little bit. Sorry for being lazy here. So the annotation colors. So basically, this is what's color coding the sample labels here. So I just made the control ones yellow and the IPF ones orange. So this is just setting those colors. This is telling it how to color code these samples, so we can easily see which sample belongs to which class. Okay, and then one more thing to copy and paste in, and this is going to be a big one, but I'm going to go through and explain what everything is doing here for you guys. Okay, so this is the command that actually makes the heat map. It's the p heatmap command. That's again this package that we're using, the p heatmap. Okay, so this thing here is just our matrix that we want to make a heat map out of. This is important here. So this is saying scale equals row. That's basically saying that all of the rows, meaning all of the gene expression across these samples, is converted to z-scores. So basically, this makes it so that we can visually compare gene to gene without, for example, like maybe in the raw data there were just some genes that just were more expressed than other genes or something. That kind of isn't the point of the heat map. We don't want to see some genes just be like solid red and others just be solid blue. We want to see the difference between the samples in terms of gene expression. So we want to normalize for the gene expression so that we can compare gene to gene. And normalizing to z-scores like this, like for every row, basically just makes it so that each gene has about a mean of 0 and about a standard deviation of 1. So that just kind of helps us like visually plot this without screwing up the visualization that we're trying to get out of this. Okay, clustering distance rows. This is just kind of defining how what kind of clustering algorithm is going to be used for the rows, and it's based on Euclidean distance. We don't need to get into that for now. That's just one method of doing clustering. Same thing for columns. Euclidean distance clustering. Clustering method complete. Just another clustering parameter. Okay. Annotation col equals samples info. So this is something you guys still need to have from the code from last time. Remember last time we made this samples info data frame? You guys need to still have that. So if you guys have started a new R session, make sure you rerun all of the code again from last time so that you can get this data frame again. Because remember, this is just telling us what class every sample belongs to, whether it's a control or an IPF class that's in that data frame there. Okay, annotation colors equals annotation colors. Remember, that's our list here, color coding the samples based on class. The controls are yellow and the IPFs are orange. Colors equals my colors. Yeah, that's this blue, white, red color scheme up here. Font size row, that's the font size of these gene labels. I had to make it pretty small because we want to be able to actually read what these genes are. So if you guys are noticing that it's like too cramped and you can't read them, try just making the row font size a bit smaller. And then font size col, that's the font size for the column labels down here. You can kind of, this is actually still a little bit cramped. Maybe I should have even made this a little bit smaller. But yeah, you can kind of read them if you wanted to. Yeah. That's just the column font size. Okay, last thing, in order to actually save this plot to a file, we just say dev.off. It's just the R way of saving the plot. Okay, let's try running this, and hopefully we get what we're expecting. Okay, so let's see if it's there. Yep, and we have successfully made this heat map plot. Okay. Yeah, so that's all I have for you guys today, basically. Just a reminder that all of the code for this series is up on my GitHub, and I'm going to put in the description a link to the code if you want to download it and try it out for yourself. The only thing that's not on the GitHub, like I've been saying, is the actual data set, just because I don't know what the rules are about like distributing other people's data on my GitHub. But if you guys need to download this data set, I recommend checking out my second video in the series where I showed you how to download it from Geo. But if you're in a hurry and you don't feel like watching that video, you can just find it at this link here. Usually with these scripts, whenever I'm working with a real data set, I like to leave a comment saying where I got the data set from. So yeah, this link here is where you can find the data set and download it. You don't need any special account or anything. Anybody can download it. It's publicly available on the Geo website. So yeah, but other than that, everything is going to be up on the on my GitHub. Okay, so thanks for watching, and any questions, just let me know in the comments. I'll try to answer them. But yeah, thanks for watching, and I'll see you guys next time.